Hello students, in today's class we are going to learn about eukaryotic translation. So here we will majorly also discuss between the differences in prokaryotic and eukaryotic translation. So coming to the steps of eukaryotic translation, similar to prokaryotes, there are 5 different stages, out of which the first 4 stages, that is activation, initiation, elongation, and termination, are the uh, main stages of translation, and post-translational modifications are basically the changes that are happening in the protein once it has been synthesized. So again, activation stage is activating the amino acids, wherein we require a pool of amino acids, 20 different amino acid transfer RNA synthetases, 32 or more transfer RNAs, ATP for energy, and Mg2 plus ions as cofactors. So the enzyme catalyzes attachment of amino acid to the corresponding transfer RNA. In case of prokaryotes, everything is a single compartment. There are no different compartments as such. So everything is taking place in the cytosol. But in case of eukaryotes, the process takes place. We have different compartments like nucleus, different organelles. So here the process basically takes place in the cytosol, where the process of translation will also take place. So the initiating amino acid is methionine in case of eukaryotes. If you remember, in case of prokaryotes, the initiating amino acid is a modified methionine, that is formilated methionine. In case of eukaryotes, we just have normal methionine recipe. The cell uses a specialized initiating transfer RNA that is distinct from the transfer RNA methionine that is used at the 5' AUG codon at the interiors in the messenger RNA. The initiating AUG codon is detected in the messenger RNA by a scanning process. So, in case of prokaryotes, we had seen that there is a interaction between the Shine-Dalgano sequence on the messenger RNA and the 16S rRNA sequence of the 30S ribosomes of prokaryotic ribosomes. But in case of eukaryotes, you don't have any such sequence, Shine-Dalgano sequence present. Rather, the 40S ribosome will scan the uh, messenger RNA starting from the 5' end. There is a modification present at the 5' end, which is known as a 5' cap. So it starts, 40S ribosome scans the messenger RNA from the 5' end till it reaches the first encountered AUG codon, which is considered as the start codon. And the reading frame of that AUG codon is also the one that is considered for the uh, sequence. Now, coming to the initiation stage. So before we discuss about the initiation stage, we will briefly discuss about the post transcriptional modifications in messenger RNA. Now, messenger RNA in prokaryotes and eukaryotes is different. In case of prokaryotes, as soon as a small stretch of messenger RNA is actually being synthesized by the transcription process, it is immediately bound by the ribosomes, and the process of translation will also simultaneously start, even when the process of transcription is not completed. But in case of eukaryotes, the process of transcription will take place in the nucleus, where all the types of RNAs are synthesized, and the process of translation will take place in the cytosol. So basically, all the RNAs have to travel from the nucleus into the cytosol, where the process of translation takes place. Now, eukaryotic uh, messenger RNA also has certain modifications that happen after transcription, and they also have significance. First modification is at a 5 prime end, and the modification is known as a capping. So this modification takes place at the 5 prime end, known as a 5 prime cap. And similarly, poly A tail is added at the 3 prime end of messenger RNA by polyadenylation reaction. So these modifications have two functions respectively. First, the 5 prime cap helps the transportation of messenger RNA from the nucleus through the nuclear pore complex into the cytosol. So basically, the proteins of the nuclear pore complex will catch hold of the messenger RNA by interacting with the cap, 5 prime cap, and it helps in transporting the messenger RNA across the nuclear membrane. Now, the 3 prime poly A tail also helps to protect or prevent the RNA from degradation by nucleases from the 3 prime end of messenger RNA. So it helps to increase the life of messenger RNA. Okay. Now, to begin with the discussion of initiation stage in eukaryotic translation process. So the process will start by binding of two protein factors to these two respective ends of eukaryotic messenger RNA. The 5 prime cap of messenger RNA is bound by the eukaryotic initiation factor 4E, okay, which is a part of eukaryotic initiation factor 4F complex. So the eukaryotic initiation factor 4F complex is made up of three different proteins: eukaryotic initiation factor 4E, eukaryotic initiation factor 4G, and 4A. Okay. So the 5 prime end is bound first by eukaryotic initiation factor 4E, and the 3 prime end, or the 3 prime poly A tail, is bound by the poly A tail binding protein, also known as the PAP protein. Here, eukaryotic initiation factor 4G, which is also part of the complex, will interact with both the eukaryotic initiation factor 4E and the PAP protein. And in this way, it basically functions to catch hold of the two ends of messenger RNA before beginning with the translation process. So this is important to ensure that the messenger RNA is intact before starting with the translation process. Then we also have another component, that is eukaryotic initiation factor 4A, which is not represented in the diagram here, but this basically has an RNA helicase activity. So whatever secondary structures are formed on the messenger RNA molecule, these are being removed with the help of RNA helicase activity of eukaryotic initiation factor 4A. And this allows the ribosome to read through the messenger RNA. So here also your 40S ribosomes of eukaryotic ribosome, the 40S subunit is a smaller subunit. So it also forms a complex with eukaryotic initiation factor 3, and through interaction between eukaryotic initiation factor 3 and the eukaryotic initiation factor 4F complex, it brings basically the ribosomes in close proximity to the messenger RNA, and that allows the 40S ribosome to start scanning the messenger RNA from the 5 prime end, that is the 5 prime cap, to identify the first AUG codon. Okay. So here is a more elaborate list of different eukaryotic translational factors and their roles. Some of these we have already discussed, and we can go through the rest in this slide. Now, coming to the elongation stage. So, in case of prokaryotes, we had discussed about three elongation factors. Elongation factor TU, that is basically forming a complex with uh, GTP, and we have a larger complex formation between elongation factor between amino acid transfer RNAs, elongation factor TU, GTP. Similar is the role of eukaryotic elongation factor 1 alpha in case of eukaryotes. Similarly, elongation factor TS in prokaryotes is involved in regeneration of elongation factor TU GTP complex. Okay, so it helps in converting elongation factor TU GDP to elongation factor TU GTP complex. So identical role in case of eukaryotes is done by eukaryotic elongation factor 1 beta gamma subunit. And the third elongation factor G, or the translocase, that helps in moment or translocation of the ribosome towards the 3' end of messenger RNA by a distance of 1 codon at the end of each elongation cycle. Identical role in case of eukaryotic uh, translation is done by eukaryotic elongation factor 2. Okay. Then in case of eukaryotic ribosomes, they do not have a specific E site. So an empty or an uncharged transfer RNA will simply fall off directly from the E site of the ribosome. Now coming to the termination, in case of eukaryotes, in case of prokaryotes, we discussed that there are three termination codons, UAA, UAG, and UGA. So similarly, there are three termination codons present in case of eukaryotes as well. However, in prokaryotes, we discussed presence of three different release factors that are involved, factor 1, 2, and 3, one of which can bind to the termination codons. But in case of eukaryotes, you have a single release factor known as eukaryotic release factor that is able to identify all three termination codes. Function is identical. It will bind the termination codon and then it will bring about hydrolysis of the bond between transfer RNA and the peptide chain, resulting in release of the peptide chain and dissociation of the entire complex so as to begin with the new translation cycle. So, in summary, protein synthesis occurs on ribosomes, which consist of proteins and ribosomal RNAs. Eukaryotic ribosomes are significantly larger. They are known as the ATS ribosomes, which are made up of two subunits, smaller 40S and larger 60S. The growth of the polypeptide on ribosome begins with the amino terminal amino acid and proceeds by successive addition of new amino acid residues to the carboxy terminal end. The initiating amino acid is methionine residue that is bound to a specialized initiating transfer RNA. The initiating 5' AUG is detected within the messenger RNA by a scanning process. Several factors are involved in initiation in eukaryotes, including the poly A binding protein, eukaryotic initiation factor 4E, eukaryotic initiation factor 4G, and so on. Three eukaryotic elongation factors include eukaryotic elongation factor 1 alpha, eukaryotic elongation factor 1 beta gamma, and eukaryotic elongation factor 2. And lastly, a single release factor can identify all three termination codons. So that's it about eukaryotic translation process. Thank you very much, and kindly like and share if you like the videos. and subscribe to get direct update on the uploaded video. Thank you.